Good evening, everybody. First of all, thank you, Dr. Caden, for such a absolutely inspiring talk, and congratulations to you and your team for reach a remarkable accomplishments milestone. Um, I would like to thank Brian for inviting me to present our work. But first of all, I would like to uh, thank Provost Bracy and Dean Miller for your support and guidance over the years. You and other senior leadership here at GW know really well what it takes to get discovery science done. So for that, thank you so much. So for the next 20 minutes, I'm going to describe briefly a story that was developed in my lab uh, purely from a discovery science perspective and gradually nudge us to the clinical side. And I think Dr. Cater presented a very good example in which we all know that tumor cells are nasty and smart, but if we are creative enough, we can outsmart tumor cells. So when you look at the cancer development and progression, you can view it as a kind of a tug of war. One side of it is cancer cells that want to invade the surrounding tissues and eventually metastasize to other organs. But on the other side is our host immune cells that try very hard to identify those mutant malignant cells and eventually try to kill them, eradicate them. Okay. But unfortunately, in many cases, cancers won the game. All right. So, but the question is why? All right. And what is the mechanism that cancer cells utilize to outsmart our host immune cells, and whether there are a way we can tip the balance? So what is shown here on the left is a cartoon that illustrates one well-documented observation in clinics, in which the immune cells have the ability to reach the tumor mass, but somehow are prevented from entering the tumor core and eventually kill tumor cells. So this phenomenon is called immune exclusion. Okay? And there are multiple possible mechanisms that could explain this immune exclusion phenotype. One of which is that cancer cell somehow has ability to build a physical barrier around the tumors. All right, that physical barrier could consist of molecule called collagen, which is normally important in our connective tissues, but somehow is hijacked by tumor cells to build this physical wall. And this is illustrated on the right side here using our preclinical mouse models. In dark dots there, brown dots there, around the edge or margin of tumors, these are T cells. Their main function is to detect tumor cells and kill them. But in this particular setting here, you can see that they are congregating along the edge of tumor, but somehow have trouble get inside the tumors. Okay, so that's a basic definition of immune exclusion in cancer biology. So there are two questions here we're trying to address here. Number one, what is the mechanism or are the mechanisms that tumor cell use to instigate this immune exclusion? And number two, whether we can find a way to overcome this mechanism so that will allow more immune cells to come into the tumor core and start killing tumor cells. So this diagram here illustrates that this so-called immune exclusion phenotype is present in multiple solid tumor types there. In the middle, you see a triple negative breast cancer. This is a cancer type that my laboratory has been studying for quite a few years. And next to it is a colon cancer. So each pie chart has three colors there, but the dark blue color represents the immune exclusion phenotype. So you can see, for example, the triple negative breast cancer, 50% of the cases actually have this immune exclusion phenotype. And if you go to colorectal cancer, more than 50% are present. So this is a widely spread phenomenon. And so if you dig deeper and say, what is really preventing immune cells to come in to the tumors? This is a technology that we utilized in collaboration with Dr. Parvatulov from the GW Nanotechnology and Nanofabrication and Imaging Center. What you see here, those cable-like structure, these are collagen fiber, okay? And little dots, maybe people in the front can appreciate, those little dots are T cells, anti-tumor T cells. And I hope you can appreciate the fact that when we look at this specific section of tumor margin, a lot of the anti-tumor immune cells somehow are stuck, okay? Embedded in those collagen fibers. This is consistent with the general idea that tumor cells have the ability to build this wall to prevent anti-tumor immune cells to come in. So our journey started by asking what tumor molecules can serve as an instigator for this physical barrier establishment. Okay. Long story short, one molecule that we nailed is called DDR1. Remember collagen I showed you in the previous slide? They form a strong barrier that prevent immune cells from coming in. And this molecule protein is a collagen receptor, okay? What this receptor does is usually expressed at high level on the surface of tumor cells. And when it binds to collagen, which is outside the cells, outside the tumor cells, that somehow triggers a signal. And that signal is transduced into tumor cells and tell the tumor cells to respond, to grow, to invade. Okay? So that's the basic knowledge about DDR1 protein. What we found is that when you take DDR1 protein away from tumor cells, that causes a tremendous effect in terms of immune exclusion. And the next few slides, I'm going to show you some of the published work. Oh, okay. First of all, I want to show you that DDR1 molecule, its level in tumor cells, in human tumor cells, inversely correlate with some of the anti-tumor immune markers. So the more DDR1 expression levels, or the higher expression levels, the lower the markers you see for anti-tumor immunity. So this would be consistent with the notion that DDR1 perhaps play a role in immune exclusion, in keeping immune cells away from tumors. Okay, so the detail is not that important, but what we did in tumor cells is to take away DDR1, which is shown in panel A. Control cells has a strong DDR1, the next lane, DDR1 is gone. Okay, so we then take these two types of tumor cells and transplant them into mice and to observe tumor growth there. So what you see in the middle panel, the red lines represent those tumors that don't have DDR1 versus the control tumors that have tons of DDR1. So first piece of information we gain from this study is that DDR1 is important for tumor cells to grow in this environment. But it's interestingly, that difference is only observed in immunocompetent mice. What does immunocompetent mean? Immunocompetent means that those mice do have anti-tumor immune cells, right? But if you put the same pair of tumor cells and put them into immunodeficient mice, the tumor cells couldn't care less whether you have a DDR1 there or not. So that gives us a clue that whatever DDR1 does there must have something to do with the host immune cells or host immune environment. And so indeed, we checked that the immune cells inside the core, inside the tumor core, what we found is that if you have DDR1 around, you don't have a whole lot of immune cells in the middle of the tumor. When you take away DDR1, you start to see those brown dots in the tumor core. So as if they somehow allow the immune cells to come into the tumor. Well, then the question, does it have anything to do with collagen? Remember, DDR1 is a collagen receptor. One of the functions is to kiss collagen and induce a whole bunch of downstream events. So again, we collaborated with Anastas, and what you see here is a high-end microscopy available at GW Imaging Center. And this allows us to look at structure of collagen fiber. So if you have a tumor that has a wild-type DDR1 protein there, you get very regular, well-aligned collagen fiber there. Whereas if you take away DDR1, which show up on the right panel here, they seem to be disorganized. Okay, so that supports the notion that somehow DDR1 has the ability to instigate this wall building or barrier building capability. And so that's the model we built based on the discovery finding there. The idea is that DDR1 somehow play a role in building up and establishing a collagen fiber that prevents immune cells from coming. So the obvious question is how do you inactivate DDR1, right? So since our initial discovery, few other labs have also shown that the conclusion we draw from the breast cancer-based work can be validated in other cancer types as well, including colon cancer. So the next question is, of course, so how do you inactivate this molecule? And for those of you who are familiar with the kinase activity, is an enzyme. DDR1 has this enzymatic activity. So you would think, okay, if you have an enzymatic activity, it might be easy to identify a small molecule inhibitor for that specifically inhibit that enzymatic activity. Except in this case, we found that DDR1 function immune exclusion is independent of that kinase activity. So just by having a small molecule inhibitor for the kinase activity is not good enough to kill its immune excluding function. So we have to turn around and find some other strategies. And the strategy we came up with is to identify antibody that specifically recognize this extracellular domain of DDR1. outside the cells being exposed, and so that it's easy to be recognized by anybody. Yet that is independent of the kinase activity inside the cells. So that is the strategy we use to develop the potentially therapeutic antibody. But let me just quickly go through the assay we use to screen antibody, because if you generate antibodies, not every single antibody you generate has the neutralizing activity. So you need some quick assay in a cell culture system to identify the lead antibody that give you the most potent neutralizing activity. So what we basically did is they take tumor cells and so-called decellularize the tumor cells. The only thing left behind is this collagen fiber from tumor cells. And we put in the middle there. And we ask whether it will prevent T cells from penetrating that fiber, that collagen fiber. So using this assay, we collaborated with leading therapeutic antibody development lab in UT Health Science Center in Houston. And I'm not going to go through all the technical procedure, but the bottom line is that through this whole process and the functional screening, we got several lead antibody that indeed can neutralize the activity of that DDR1 molecule. Let me just take you over several pieces of key evidence here. Remember the tumor study I presented earlier? What we did is treating the animal with the antibody. And in red is the antibody-treated tumor growth. It is significantly lower or smaller than the control group there. And interestingly, this is only true in immunocompetent mice, not in immunodeficient mice. And again, that goes along well with the idea that DDR1 in this particular setting play a very important role in immune excluding function. If you take away the whole immune system there, you don't expect to see much difference. And again, the antibody treatment would then increase the infiltration of the anti-tumor immune cells into the tumors, which is shown in red compared to the control. Last but not least, when we use this fancy microscopy to look at the collagen fiber structure, so what you see here is that if you treat tumors with anti-DDR1 antibody, it indeed disrupts the collagen fiber structure in a similar fashion as the DDR1 genetic knockout, as I shown you earlier. So that's basically the idea that you inhibit DDR1 activity and sort of melt away that physical barrier that separates tumor cells from the immune cells. In the last few slides, I want to show you some of the efforts we made in collaboration with commercial companies to push it to a clinical trial. Now, this is a bit tricky point here. You have antibody that work in mice. How do you move it to humans? And there's a trick here called humanization. I'm not going to get into detail of that, but just make the antibody more acceptable by humans, right? And then, of course, we need to prove that the same humanized antibody work in tumor inhibition. And that's shown on the right graph here. And the red is antibody treated versus the control group there. So the humanized antibody is still effective in tumor inhibition. And so just want to use this chart here to illustrate a few milestones here. My group moved to GW in end of 2018. We received our first DDR1 related R1 grant from from NIH, and get our story published at end of 2021. But as you notice, between 2018 and the 2021, we didn't have any federal or extramural funding for this particular project. What we did have, start resources funding from School of Medicine, from the Provost Office. So I'm very grateful for your support to help us jumpstart this project there. And so I'm not going to go through all the individual steps here. I eventually, with the help from the Brian's group, we filed a patent in partnership with the UT Health Science Center in Houston, and identified a company that licensed our patent and helped the company to raise some VC fund. And then very happy to see that the company took over and pushed the antibody all the way to phase 1 clinical trial. And so these are my disclaimer here. I currently have no role in the company, and all this public information from the company website there. So it's the phase 1C trial is still going on, and the company is planning to launch a phase 2 later this year. So as a basic scientist who have spent multiple years in laboratory research, what did I learn from the whole process? I'm sure that this is all obvious to you in the audience, but to me, my lessons are the following: talk to TCO as early as possible, right? As Brian said in his slide, and to me, collaboration, especially cross-disciplinary collaboration, is very important, because for this project to reach to the clinical trial, as you can imagine, we have collaborations with antibody experts, with clinician scientists, and with even experts, in-house experts on microscopy. And finally, I think a little bit adventurous spirit is important, that allow us to learn new things even when there is a high risk of failure. Thank you very much. Well, good evening, everybody. First of all, thank you, Dr. Caden, for such a absolutely inspiring talk, and congratulations to you and your team for reach a remarkable accomplishments milestone. I would like to thank Brian for inviting me to present our work. But first of all, I would like to thank Provost Bracy